Okay, good morning, everybody. It's just a few minutes past 10, and welcome then to the 8th talk in our series here. And I'm Stephen McLaughlin from the MRC Laboratory of Molecular Biology, and today I'm going to talk about Biosensor Technologies. So what is a biosensor? Well, today we're going to talk about a range of instruments where the biosensor entails you're looking at interactions between macromolecules, where one of the macromolecules is going to be attached to a surface. We're going to call that the ligand. And then we're going to flow across its interacting partner, and we're going to call that the analyte. And so all these different biosensors then use different physical changes at the surface to measure the interactions. So why would you kind of want to measure kind of interactions between, two macromolecules? Well, one thing might be because you want to see, well, do they interact, do they not? You might want to say, well, how strongly do they bind? You want to find what the dissociation constant is. You may want to probe the interface by doing mutational analysis and seeing how that affects the dissociation constant. You might want to look deeper in and have a look at the on and the off rates and to understand the dynamics of the system. And with the switch sense, then we'll also look, you can do determination of the size of the interacting partner. So what's the relevance of kinetics? Well, as you can remember from Chris's single molecule talk, the cell is very dynamic, and there's also macromolecules, and it's also very, very dynamic. So, when we talk about binding constants, then that binding constant really is determined upon the underlying individual on and off rate constants, just by a simple ratio here. Now, on rates, they're going to be, because it's two molecules coming together, then they're going to be concentration dependent of the observable rate. So, the binding level is just not related to the affinity. It's quite important, of how much of each of the reactants are there. While the off rate is not really dependent upon what the concentration of that is. But it is related to the half-life of how long this complex stays together. And as we'll see later, then the same affinity can be a result of completely different kinetics of on and off rates. Okay, first of all, there's a whole range of biosensor companies out there, instruments. And in fact, yesterday I kind of added a few more on there. And some of them have got used the same kind of physical principles, like SPR. Others have got different types of physical properties underneath. But I'll just concentrate on 3 today. These are the ones that we have at the LMB. It's important to understand the principles of each of these 3 things, because that will be able to relate to any other kind of biosensor that you use in terms of what the controls are and what the problems could be and how you would go about analyzing biosensor data. So first off, let's have a look at the biocore. Here's the T200, and what we have, the biosensor is a glass slide, and there's a very, very thin layer of gold. And in the standard kind of CM5 chip, then there's this dextran layer on the surface. And it's formatted in this little kind of plastic cassette, which you can insert into the instrument here, and then microfluidics kind of clamp on to make 4 channels either in series or in parallel. And so that we have then buffer flowing through over the surface of the biosensor. And then on the back side, then there is a prism, and then optics will look through to see what's happening at the interface here. For the octet, then we have then individual sensor fibers where you have a biocompatible layer here, and where you can immobilize molecules. And the light is then shone directly through the sensor tip. And these all come in nice little racks here of 96. And so it's formatted in, say, either 96 or 384-well format here, where the sensor tips are then in this optics box, and then they dip into the solution with all the reactants here. And then it's white light that's actually interpret what's happening at the center there. So with switch sense, then it's slightly different here. You have this chip where there's 4 channels. And in each of these channels, then there's lots of electrodes coming off. You can see these gold things here. And if we kind of look up, then there's 6 microelectrodes. And if we expand up again, in each of these microelectrodes, then there's DNA nanolevers then that are made onto the surface. And the DNA strand that's attached to the surface then is a fluorophore. And we're going to measure then what happens at the surface by fluorescence. Okay, for detection in SPR, we have light incident polarized light is shone on through this prism. At an angle, we get total internal reflection, but you also then get then light absorbed onto the surface here. On this gold surface, you need it because then there's surface plasmons. And so at a certain angle, then the kinetic energy then of the incident light then matches the energy of these plasmons here, and that causes them to move and then resonate. But of course, you've taken energy away, and so at a certain angle, then there'll be a reduction in the reflected light here, and this is what's called the SPR angle. Now, where this angle is really will depend upon the refractive index at this interface, and which will then also depend upon what's bound on there in terms of the mass. So as, say, we flow something across that binds, the SPR angle changes, and we detect a mass change. And then as we flow buffer then away, we get dissociation, then the angle goes back. For octets, it's also called Biolaryferometry. And so what happens is that light is shone through, and then that gets reflected back either from the surface or from a control layer here. And these then waves then interfere with each other to produce an interference pattern. And you see across a long wavelength here. And so if anything then binds onto the surface, this is going to change how this light is reflected back. And so you get a change in the interference pattern, and there'll then be a change in the wavelength where these maxima and minima occur. And so in these biosensors then, because they are in 96 well format, there's different formats here, 96 well, 4 well, or tilted well. You can use these volumes here. Then you can look at, say, binding on a ligand onto the surface, and then taking them out, and then putting them into, say, where an analyte is, and then looking at the interaction, and then putting them into buffer, and then looking at the dissociation. For switch sense, what we've got is we've got an LED, and that is then through an objective microscope lens is then focused onto one of the electrodes. And then if there's any fluorescence, that comes back through this dichroic mirror and is counted. And of course, the electrode then is switching and for positive and negative with this frequency generator here. And that's then linked to an event timer to correlate both the fluorescence and the time of switching. So that when the surface is positive, DNA is obviously negative, that's attracted, and the fluorescence of this fluorophore that's attached onto the nanolever is quenched. If you then switch to the surface being negative, that will repel the DNA, the nanolever will start to stand up, and you'll then get fluorescence coming back. And then this will cycle backwards and forwards. And so we can measure the fluorescence counts here, and you can see that we're looking at the dynamics then in the microscale second. So that the frequency might be 10,000 kilohertz. And another thing that we can actually do is we can actually look at how long it takes for the dynamics of this lever to kind of go backwards and forwards. So that if, say, something binds onto the surface, you'll get frictional drag, and so it'll be slower then to move. And you can then use this to determine sizes by looking at the difference in the dynamics. And here's an example here of just a biotin addressed nanolever, And then when you put the antibody against biotin, then this slows down the dynamics of it switching. There's also other modes as well that switch sense can use. Okay, we've looked at dynamics, which we can then look at, binding and diameter changes, conformational changes, melting. We can also look at proximity sensing. So if you remember from the fluorescence talk, then, with the MST, we talked a bit about that in terms of changes of the fluorescence properties of the fluorescent group with something else bound close by. And in this case, you don't need to switch. You can just have the nanolever standing up. And if something binds, close by to the end of the nanolever, then this may change the environment of the fluorophore and changes in fluorescence. And again, we can use that for binding and kinetics and also melting. Now, another thing that you can do with the switch sense, again, of course, it's a DNA nanolever, and you can use that to then look at DNA binding proteins, particularly like nucleases and polymerases, where you can have a half-formed, say, nanolever, and then you can look at how this binds, and then when it starts to then make either make nucleases on, so then you get a stiffer nanolever as the duplex forms, or alternatively, you could look at nucleus activity when the ends are trimmed back. And that will then change the flexibility of the end here, and then what the total fluorescence is going to look like, because you'll have the fluorescence group coming closer to the surface or further away from the surface, depending upon what the state of the nanolever is. Okay, so overall, if we're kind of looking at kinetics, then in biosensor, we tend to get these kind of what we'll term sensograms. You'll have a phase where you're looking at association kinetics, and if it reaches an equilibrium position where the on and off rates are equal, then there won't be any change in the signal at this time point. And then, when you start to flow buffer through and get dissociation, then you'll see the signal going down to get dissociation kinetics. So the units, well, what units to use? Well, SPR, we call it resonance units, and that's equivalent to a mass bound onto the surface. So typically 1,000 units is equal to 1 nanogram of bound protein per milliliter squared, where 1 resonance unit is equal to 1 × 10⁻⁴ degrees change in that SPR angle. In the octet, the BLI, we're going to get these units of nanometers. Now, that's not size at the surface, it's actually a wavelength shift of that interference pattern. With switch sense, we're looking at the nanolever dynamics, and we'll look at the rate of fluorescence recovery and the dynamics of switching, so it's called the dynamic switching units. Right, so there's different types of biosensor. Which one do you kind of choose for what experiment? Well, obviously, the SPR and the BLI can do, and switch can do affinity and kinetics. Switch sense has got an advantage of looking at DNA enzyme kinetics and conformational changes. In terms of dynamic range, well, in terms of mass, the Spr has really no limit. There's a set lower limit than for the BLI. Switch sense, I'm not sure what the limit would be on that. In terms of affinities, then Spr and the octet are pretty similar. While switch sense, then you can go to much lower affinities, and we'll see why that is in just in a second. And that's because of the difference in how it can look at on and off rates. So with octet and with the switch set with the SPR, then they're pretty much the similar kind of range of association rates, while the switch sense can then look at much faster rates. And conversely as well, then in dissociation rates, the switch sense can look at much slower rates, because you can look at reactions dissociation rates for a much longer time scale, and so then consequently you can look at a much broader range of tight affinities. So temperature control is quite important for if you're looking at kinetics. the Biocore has got a very, very good temperature control. With the octet, it's because it's plate-based, it can go from ambient then upwards. You can't really cool it. And the switch sense then is similar to the Biocore, and you can actually go to much higher temperatures. In terms of throughput, well, Biocore can do 96 or 4 well plates, similarly to octet, and with switch sense you can do 96. And it's only really the switch sense you can do sizing experiments. So what are the kind of issues? Well, I mean, one thing would be the cost. I mean, if you use Biocore, then you're going to use chips. So, there's a certain cost per experiment. While with sensors, because they come in 96, then you can maybe only use 1 or 2 rows for an experiment. In all of these biosensors, you're making a surface, and so how is that going to fundamentally affect how the interaction occurs? If you bind something to a surface, are you going to get steric hindering of that interaction, or are you changing the entropic changes upon binding? There's obviously always an issue of non-specific binding. You've got a surface, so will your binding partner bind onto that surface and basically swamp out any positive interaction with the ligand? Buffers, make sure you've got a buffer that actually will work. We'll see later on that Biocore is really sensitive to refractive index changes, and switch sense, given its electrical switching, is sensitive to salt. And okay, the detection limits, as you've kind of seen with the switch sense, you can look to very, very fast on and off rates. And so obviously, for if you're using the other instruments, there'll come a point where really you won't be able to measure that rate, because the response rate of the instrument isn't really fast enough. But we'll see how you can analyze that fast on, fast off data later on. And we'll mention that again, there's fitting issues. If you've got very, very slow dissociation rates, that sometimes can be very, very difficult to analyze. Okay, so let's think about doing some biosensor work. This is kind of the workflow of things you kind of got to think about. Okay, first off, good samples, make good buffers, make sure your buffer is filtered. Is it compatible? If you're going to cross-link via amines, as we'll see later, make sure you avoid tris. Do you have to include detergent or DMSO? And for SPR, it's really important that you match the running buffer with the buffer that the analyte is in to minimize this bulk refractive index change. For samples, you don't want to put on aggregates. Make sure they're nice and clean. You're going to measure kinetics or binding constants, so you need very, very accurate concentrations. And the big question, obviously, people will say is, okay, you've got an analyte and you've got a ligand. So which one do you attach onto the surface? Which one do you flow across? Well, in an ideal world, you would do it either way to make sure that the binding constants or the kinetics are the same, whatever way you do it. But in reality, there may be issues such that you can't get one of your components to a high enough concentration, or it has non-specific interaction with the surface, so maybe that's going to be a good one to kind of couple. Or it could be valency, so that if you want to look at a specific interaction, avoiding kind of affinity issues, then you'll attach something to the surface. Okay, how much do you put on? Well, for SPR, then there's kind of a rule of thumb, which would be, I mean, it used to be like, say, putting on enough ligand to get 100 RU maximum response. Nowadays, they're kind of saying, put on a bit lower. And so how do you know you're going to get, say, this kind of maximum response of about 100? Well, I mean, that will determine, by, how much the size of the ligand and the analyte is, to get this maximum response, how much you're going to put onto the surface of the ligand. So you can imagine that if you had a very, very small analyte, say, a small molecule, so then that's going to be, much, much, much smaller than, say, an ordinary protein. So to get a good response, you're going to have to put quite a lot of the ligand on the surface. Conversely, if you had, say, a very, very big analyte, then you only really need to put on a small amount of ligand on the surface, and you'll get a big response because you're putting on a big mass. For BLI octet, well, I mean, because everything is running in parallel with these sensors, you can just do a kind of a preliminary experiment and say where you can have the ligand here in different concentrations, and these just buffered here, and then have the analyte at a concentration where you think you're going to get a good response. And then you can just then test out, well, how much do I need to put on? How much do I need to put on to get a reasonable response? And obviously the lower the better, but a typical thing might be, say, getting 1 nanometer of say ligand onto the surface over about 5 minutes. For switch sense, well, you've got a set amount of nanolevers on the surface. So we then put on, say, about 100 to 500 nanometers. Oh, sorry. We put on about 100 to 500 nanomolar of DNA. If avidity is an issue, then we can do this. We can put on a lower level. So and we can reduce the number of say nanolevers either by electrical desorption or by using different chips which have high, different densities. So of nanolevers on the surface. So in this case, then you're going from 50 nanometers distance to 100 nanometers distance to the very low, where you're 200 nanometers distance between the different nanolevers on average. So how do we get a ligand onto the surface of the chip? Well, for SPR and BLI, then there's 2 methods. You can capture it by using a capture molecule, which of course could be like biotin or an antibody or GST or nickel or an antibody, or you can chemically cross-link. So you attach the ligand on with a cross-linker. So, by amine or foul coupling. I mean, obviously there's advantages and disadvantages. The advantage here, if you do a capture molecule, with the exception of streptavidin, that, during different cycles, that you can just strip the ligand off after you've measured, one interaction at one concentration of analyte, and then recapture again, and then measure a different concentration of analyte. While, with, say, the cross-linking, then one of the advantages as well, you've put the ligand on, once, and that's all you need. So you need very, very low amounts. But of course, as we'll see later, that sometimes you can destroy the ligand on the surface, and then you have to start all over again. Okay, so what kind of chips can you use? Well, there's a standard chip, which is a CM5 chip, and then there's different types of dextran chips here for different kind of types of experiments, such that you've got very, very large analytes, or you need a much denser dextran layer to get for low molecular weight analytes, or alternatively, there's a PEG one that they've brought out, which is good for very large and multivalent binding partners. There's various chips that come with affinity tags, so for biotin ligands, or for ones that can bind onto lipids, or this is the HIS. And again, there's other ones which will capture antibodies. It's very similar then for octets. There's amine or hydrophobic ones, and a whole range of different affinity tags. And also you can see there's a whole range then of also antibody capture ones. And this kind of emphasizes that both SPR and octet are used in industry and certain biopharma for development of antibody-based therapies and biosimilars. So, if you don't come across this in your academic career, you move to industry, then you'll definitely come across these techniques. So now, switch sense. Again, you've got to then make a complementary strand to the nanolever. That's the strand that's attached onto the surface. And so you can then say amine couple or thiol couple or click couple, or you could buy complementary strands that have got affinity capture tags on there. Again, there's various ones here. And again, similar, there's antibody-directed ones. And there's special chips, which would be for bifunctional antibodies or for enzyme polymerases and nucleases. Right, so for amine coupling, what we would do would we put a crosslinker on the surface here, which is then going to generate a good leaving group for then a protein coming in with an amine group, typically lysine, is going to form a crosslink here. For file coupling, then we then generate the surface with this PDA, and then that's going to be reactive against cysteines. There's an alternative maleimide, which will then be a permanent capture. So how do we get something onto the surface to amine couple? Well, we can take advantage of the fact that the dextran layer here is slightly negative. It's got also for carboxyl groups. And so that if we inject a protein in below its pi, then it's going to become slightly positively charged and going to be attracted onto the surface. And so, of course, if it's attracted onto the surface, then it's more likely that it's going to be come in close contact with the crosslinker and be cross-linked. So there's a little kind of wizard in the software where you can then kind of inject in your protein at different pHs here in acetate, and then you look at the response here, and you can see really looking for linear responses, so that you can kind of then say, well, if I inject in for this amount of time at this pH, then hopefully I'm going to attract about this number of units onto there. So a typical kind of reaction would look like this. So you've got amine cross couplers going on, then you inject in your protein at a pH below its pi, it gets attracted in, and then you stop, and you can see that, well, not all of it then gets cross-coupled. It's a bit of a drop there. And then you block any kind of free cross-linkers then with ethanolamine. And then you end up here with this amount of immobilized ligand. So other types of chips would be, say, with the NTA. So that's just essentially like a nickel column, where you then attach on then nickel, and your hexahis protein comes on, and then it's just attracted onto the nickel. With streptavidin, then the chip already comes with streptavidin attached to the chip, and so then anything biotin-related ligand will come in, and then that will be permanently attached on there, and then you can flow across your analyte. For looking at membrane proteins, and there's two types of chip. Here, there's a hydrophobic surface where you can layer on a lipid onto the surface. And for looking at liposomes, this is one chip, which is has a molecule here that will integrate into a membrane and then capture these liposomes. Then you can then look at any binding of analyte onto, say, a receptor that's in that liposome. Okay, so for switch sense, it's slightly different. We want to form this nanolever. So there's one with the fluorophore that's permanently attached, and we want to flow across this one in blue, which is going to have our analyte. So we need to get this complementary strand then attached onto the ligand. So we could do that with, say, amine cross-coupling, or with file reactive groups, or click chemistry, or we could just then, as I said before, then use different capture tags that are already on this surface. So there's different types of chips then in switch sense. So we have, say, multi-purpose one, which either comes with 98. Sorry, it either comes with 48 or 96 base pairs. And using the 96, then if you've got something that's quite large, then you want that to be a bit further away from the surface, so you don't disrupt the kind of switching. And with all the purpose chip, you can do all this kind of range of different types of experiments. If you want to look at enzyme reactions of DNA polymerases or nucleases, then you need a special enzyme chip. And if you're looking for bifunctional chips, say, for biospecific binders, if you're looking at antibodies, then there's a special chip for that. And the company then has developed even newer technologies now, such as DNA origami, but that's on the more newer instruments, which we don't currently have at the LMB. Okay, so you've got your ligand onto the surface. Now let's see, how you're going to kind of do an experiment and make sure that it is actually something that's worth analyzing. Well, you're going to look at controls. You're going to ask yourself, is there non-specific binding? Do you need to regenerate the surface to look at different concentrations? And then, go around again, see, can you get a good experiment to look at kinetics and binding? Well, in terms of controls, then with the SPR, you've got four different flow channels, and you can run these in series, either 1 and 2, or 3 and 4, or completely in series here, 1, 2, 3, and 4. So if we say make the first channel either channel 1 or channel 3 as a reference channel, and if we treated it as as the ligand addressed channel, then we can use that as a reference. So a good reference might be, say, a biomolecule that kind of mimics properties of the ligand, say it was a mutant that didn't bind, or a bit of random DNA. And the reason why we do that is we want to take away any kind of this bulk refractive index change or any kind of slight non-specific interaction that you kind of see from the signal from the ligand address channel here, to actually get what the real signal is. So here's an example here using a GST capture molecule. It's an anti-GST antibody on both the flow channels 1 and 2. And so as a reference on 1, we'll put on recombinant GST, or that's addressed singly on there. And then on flow channel 2, we're going to put our ligand of interest that's being fused onto GST as a recombinant protein. And then we will flow the analyte over both surfaces. And as you can see, here there's a bit of non-specific, binding onto the surface here. And we also then do an injection of buffer as well to see if there's, just any kind of drifts or just, signals there. So that when we take away the reference channel, then we can get a nice data here that we can actually fit. And as you can see here, then these are all different cycles at different concentrations of ligand, all overlaid on top of each other. And so at the end of an interaction, what we do is we regenerate the surface then by injecting a low pH, which will then break the antibody GST interaction. So we'll get back down to that baseline level of where we start, and then we'll do another round of capture, interaction with analyte, wash off again. So then we can build up a complete pattern of this interaction kinetics at different concentrations of analyte. So regeneration, for other systems, then it could be that you use salt or acid or alkali or detergent, and so hopefully what you'll get is you'll regenerate and you'll get back down to baseline. And so when you inject in another concentration of analyte, you'll get a good response. You don't really want to get it where your regeneration is that you don't get back to baseline, because you really then won't get a good response. Of course, what can happen is that you inject in your regeneration solution, you get down to baseline, that all looks good, but then your second response is poor. And of course, what that means is, of course, you've then destroyed the integrity of your ligand that's attached onto the surface. And if that's chemically cross-linked, then, well, you've just got to start again with a new set of channels. If this is a problem, on the biocore, then you can do single cycle kinetics, and obviously in the octet BLI, your sensors are all in parallel, so you don't necessarily need to regenerate. Okay, so for single cycle kinetics, instead of, say, doing a whole load of different concentrations and regenerating in between, what you do is you do a series of injections of increasing concentrations of analyte, and then only dissociate at the very, very end. And in this kind of test example here, between multi-cycle kinetics and single cycle kinetics, we essentially end up really with the same kind of biphasic KDs here. Right, so we look as if we've kind of got good binding, but another thing we need to think about is mass transport, particularly in Biocore. Because you've got this dextran layer in here, then it could be that diffusion into this layer could become limiting, such that the flow can't replenish the amount of analyte going across the surface. And during association phases, or in dissociation phase, what could happen would be is that you get dissociation coming off, but really the bulk flow is not really fast enough such that then you get rebinding onto the surface, and so the dissociation looks much slower than it should be. Now, an issue of that is, of course, that this rate here of diffusion across is then depending on the cube root of the flow rate. Well, how do you tell you've got a problem? Well, if you look at the analysis of different flow rates, you can see here in this case that, well, the same concentration on analyte, different flow rates, and we're getting different kinetics, as opposed to here where there's no mass transport. So how to overcome it? Well, use high flow rates, use a low amount of immobilized ligand. You can fit the data and include mass transport variables in there. But obviously, if you don't need to invoke it, then don't use it. Okay, and in octet BLI, you can use controls basically within these parallel sets here. So you can imagine here's the ligand, where we're going to then this is going to be the reaction where you're going to address the biosensors, and you could have, say, one here where you don't have any ligand in here. And when you come to measure the analyte, you could have one of the sensor where you don't then have any kind of analyte in here and just buffer, so that when you load, here you go, you're loading here, and then one of them isn't going to be loaded. And then so with a ligand, and so you can look at what the background interaction is of the analyte to the surface. And here in this one, which is this one here, G, is that you've got no binding partner, and then this is just looking at really the instrument drift. With switch sense, with the one that we've got is the RX2. It's a 2-color system. So there's 2 different types of nanolever on the surface with different sequences. One which has got a fluorophore that which fluoresces in the red zone, and one which fluoresces in the green zone. And so you can imagine you could have an internal control where you attach one complementary sequence that will attach to the red type, so that your ligand will bind there. And then you just flow across a non-addressed complementary strand, which will go to the green ones. So then you could look at non-specific interaction with the nanolever itself. Okay, so how do we, if we do get non-specific binding, then what can we do about it? Well, you could change the chip. You could change a different type of dextran chip. You could use a PEG chip, or you could say cross-link the surface with PEG. Change the running buffer, the pH, change the salt concentration. You could put in additives such as detergents, tween, or there's blocking agents such as casein, PEG, gelatin, or BSA. Maybe EDTA might help. And Cytiva then sell a non-specific binding reducer, which is just soluble carboxymethyl dextran, and you add this in at this kind of concentration. For streptavidin chips, then you could maybe block with biotin or biocytin. Okay, so everything looks good so far. You've got good signals, you controlled well. So let's do some kinetics on binding. Well, for kinetics, at least 5 concentrations of analyte. For affinity, you're going to need a lot more to go through the KD. You need an adequate concentration range above and below the KD for affinity. For kinetics, you just need concentrations where you've got a good range of signal, such that you can then look at the association and dissociation rates. Do things in duplicate and triplicate if you can, Sometimes things go slightly wrong, might be an air bubble or something, and it's always good to then say, well, you can discard one concentration. Obviously do, include buffer controls as well, because you need to be able to take away kind of background drifts and bumps. So what data are we going to look at? Well, that really depends upon the underlying, kinetics. So let's kind of imagine we've got something that's very, very tight. It's got 10 nanomolar binding affinity. Well, that could be made up of completely different association and dissociation constants, as we'll see here. So in blue here, we've got something that's very, very fast on and very, very fast off. And in, say, yellow, we've got something that's really reasonably, fast on, but much slower off. And in red here, we've got something that's very slow on, but it's actually really, really slow off. And you can see that, well, something that's really slow off is going to bind onto that receptor or protein for a much, much longer time, while something that's very, very fast off, it's basically gone very, very quickly. And why that might be important is say, if you're looking at, say, drug screening, and here's a whole array of different hits from a screening campaign, where you got log K on and log K off here. And then these parallel lines are lines of the same affinity. So imagine here you've got compound 1 and compound 2. They've got pretty much the same affinity within the kind of same range. But if we look at compound 1, the kinetics are slow on, slow off, while compound 2 is then much, much faster on and much, much faster off. So in terms of what's a good drug and what's not a good drug, well, perhaps compound 1 might be better to develop because it's got better pharmacodynamics. It's going to bind onto that target for much, much, much longer. Okay, so this is the kind of data that you might get at extreme, then fast on, fast off, and then slower on, slower off. So in slower on, slower off, you're not going to reach this kind of equilibrium position where it's flattening off. So you're going to need to look at the individual kinetics. While here in fast on, fast off, well, the response time of the instrument isn't fast enough to look at the kinetics. So you're going to have to look at this equilibrium position. Okay, so you're going to then, to fit the data, you're going to subtract your reference data and channel, and then double reference by then subtracting the zero concentration of the buffer. There's different software for analyzing that comes with the instrument. So for Biocore, then there's the BioValve and the T200, but you can always then take the data out and then fit that in things like Prism. For SwitchSense, then we're looking at kinetics. And again, they sell Switch Analyze, which comes with the instrument, and you can analyze the data on the instrument and to get binding constants off. And also as well, they have a software then to fit then sizing information as well. In this case here, it gives a diameter of what the analyte is. Okay, so let's look at binding data. Okay, in steady state, then we're looking at this equilibrium position where it flattens off. And we just take what the response is, plot that against concentration, and then we can fit this to an equation here, where this is the concentration of analyte, this is the maximum response that we got here, and this is the association constant, which is then just 1 over the association constant. Now for kinetics, then things get a bit more difficult because we need to find what the binding constant, dissociation constant is here, and what the association and dissociation constant is in this phase. And you'll find it in the programs, and there's lots of different kinds of models that you could possibly fix by which model you fit. Well, we'll come on to that and in a minute or two, and also in the lecture on data fitting. So if we imagine here's the different sensograms, we're going up in concentration, and we want to find what a binding constant is. So we need to find what the individual dissociation rate constant is and the individual association rate constant. So the dissociation, as I said originally, then the concentration of the complex formed, what the dissociation rate, it'll be all the same, no matter what the concentration is. So we can fit all these ones to get then the same kind of KD out of their dissociation rate constant. Now, in this phase here, where we observe the association, the shape of the curve then will depend upon what this association rate constant is, but also what this dissociation rate constant is here. And so we need to put those both into the equation. So we need to find what this dissociation is first. So we will input that into our data fitting. And so here's again, the association rate constant. This is the concentration of analyte. And we can fit this in against the time. And of course, the Rmax is then the maximum response, which you may not necessarily see if it doesn't get up to equilibrium. So one issue can be is that, well, if you get this wrong and you get a bad fit, then that will propagate itself into the fit of the association rate constant. And as we'll see in a second, then this will end up with the wrong KD. So in this case, then you see it was very, very difficult to measure what the dissociation rate constant was. And that might be because was it very, very slow? Or was it because there was a bit of drift in there that we couldn't really account for in the double referencing? And so in this example, then I measured it and ended up with a KD which was very, very tight, which, was that right or was that not? Well, here's another way to kind of fit the data. We can take advantage of the fact of this association phase. The observed rate constant then depends upon both the dissociation and the association rate constant. So if we fit the observed rate constant, it's just a very, very simple exponential, and we do this at different concentrations, then for a simple one-to-one interaction, it should be a linear dependence upon the analyte concentration, where you can see that the gradient of that line will depend upon the association rate, and the intercept then with the y-axis here will depend upon the dissociation rate constant. So if we just fit that, and we can then get individual rate constants, and we divide those to then get our dissociation rate constant, which in this case came out about 1.8. And that very much matched what the binding constant was determined by ITC. Okay, so you do analysis, and then, okay, well, what's a good fit? What's a bad fit? Well, one thing is to say, well, try the simplest first. Is it a good fit? Well, look at the residuals. Is there something happening here? What are the errors on each of the individual parameters? What's the chi-squared, which shows you kind of how different the fit is to the actual data. Now, it'd be obviously very easy just to kind of say, well, okay, this isn't fitting very well. Maybe I'll try a very, very complicated model. Oh, look, my fit is better. Oh, therefore I've got it must be this complicated model. But you've got to ask, well, is that actually relevant? Are you just invoking a complicated model because it fits better, or do you have poor data? So here's in the last kind of 10 minutes or so, I'll just run through some examples of how we've used these different biosensors on different projects. So the first one here was a few years ago, and that was looking at the activation of this APCC E3 ligase, which is very important in cell cycle control. And so this is a huge complex then that gets activated then by binding onto this cadherin 1. It gets activated then, so then it can then bind onto its E2 ligase. So we wanted to know really, well, what happens then? What's the difference in affinity between the APO and then the activated form? So the way we did that was then take the E2 ligase. It's quite small. biotinylate that, and then attach that onto a streptavidin chip, and then flow across both the apo and then this activated ternary complex. So here is a streptavidin chip, and you can see you can just then inject it in the ligand and look at what kind of the response is until you kind of reach a level where you think, okay, that's where I really want to be. Then I did a test binding, and what I found was, unfortunately, that the analyte, this APCC, when it actually had quite a big non-specific interaction with the surface. So needed then to look at conditions to try and take that non-specific binding away. Just trialed kind of different detergents or BSA or different salt to find kind of conditions where I could reduce that non-specific binding down to an acceptable level, which I did. And then I was ready to go. Okay, and so setting up an experiment with these instruments is very, very easy because it's all computer driven. There's lots of wizards and templates where you just need to really fill in what you're going to add in. In this case here, this is just a simple, I got a sample and I'm going to do a regeneration, and I'm going to contact the analyte for 120 seconds at a certain flow rate, and I'm going to dissociate for this time, and then I'm going to regenerate with this solution for this time. And I'm going to put in all the different concentrations of analyte. I'm going to flow across. It will then tell you, the instrument will tell you, well, this is where I want you to put all the solutions in. And you hit go, it'll just run overnight, and then it will stop. And you can then get these kind of data, which you can adjust. And you need to take away the reference, and then again, double reference, taking away the buffer control, and then you can fit the data. And in this case, I could compare the results then of the individual kinetics here in red between the apo and the ternary. And I could look at the equilibrium response as well at the end. And I could also look at the observed association rate constants as well by looking at the observed rates against concentration. And from the individual rate constants, I could then look at the kinetic binding constant and then compare that against the equilibrium binding constant from this equilibrium data here. And they all match very, very nicely. And we could see that really upon activation, then there's a much tighter binding of the APCC to then its E2 ligase. And that is mainly through changes in the off rate. So here's an example of doing kind of ligand screening. Obviously, this is small molecules in say 96-well format being then screened across, say, something that's attached to the surface. Okay, and if you remember from before, then to get a good response, you need quite a lot of ligand attached onto the surface. And because these small molecules are going to be quite small, then you get quite a small response. And typically then all these molecules are going to be in a library, and they're going to be dissolved in DMSO, and you're going to need DMSO in the running buffer as well. And because DMSO is hydroscopic, then not all DMSO is the same. If something's been lying around in your cupboard for a few months, it's going to have water in it, and so it's not going to be 100% anymore. And similarly, it may be that in a screening library, there may be slight differences in the DMSO concentration. DMSO concentration changes will cause bulk refractive index changes. And so then you've got to disseminate, what's a bulk refractive index change and what's actual real binding. So we can take care of that in the biocore then by doing a solvent correction, by injecting a whole series of different DMSO concentrations and then correlating those changes and correcting for them. And so here's a whole screening, and it's always good then to have, if possible, to have, say, negative controls, which don't bind your target, and positive controls, so that you can then kind of set kind of limits about what you think your signal is going to be, so that when you analyze then the small molecules, you can kind of go, well, these ones here really aren't binding. These ones here look as if they're actually binding. And then there's these ones here, which look a bit strange. And so you can rank the binding, and it's all kind of built-in software. So for octet, then here's an example I did with a COVID project, and we wanted to look at then the spike protein. It's trimeric, it was soluble. I wanted to look at binding to the human receptor ACE2, and that was functionalized then with an FC, which will then bind onto a protein A sensor. And so here we go, we're loading it up, and so there's the ACE2 binding, and then we dip that then into different concentrations of the spike protein here, and then look at dissociation. And because it was protein A, then we could actually regenerate the surface then by low pH, get down to baseline levels again, and then take the same sensors, but this time not addressing them with ACE2, just putting them into buffer, and then taking the sensors and putting them into the spike protein row here to then look at what the non-specific binding is, to be able to then correct for any non-specific binding. We could also need to correct for these little jumps here, take away the buffer, and then fit the data to find out what the binding constant is from the on and the off rates. And I think it's really kind of sobering to kind of think about the fact that, you know, when this spike protein actually binds onto the ACE receptor here, it doesn't come off. It's very, very slow off rate here. And so that kind of rate means that for half of the spike protein that's bound to come off, it'll take about 5 hours. And that's because you've got a trimeric spike protein and you're looking at a big avidity effect. Okay, so for switch sense then, a slightly different format where you need to then really flow across your ligand, either a capture molecule or your ligand that's been attached onto the complementary strand of the nanolever. You then generate duplex and then flow across then your analyte on top. And at the end, you can completely strip the duplex by using harsh conditions. Then that will break the duplex, and then you can wash off your ligand and then go through the cycle again. Here's some very, very quickly. I'll just do 2 examples. Now here was one from the Passmore lab where they had this RNA binding protein here, which had this huge low complexity region here, and they kind of wondered, well, okay, well, what's that actually doing? And what's this region here that seems to be quite important for the interaction? And because it seemed to be that it actually increased the affinity if you had this upstream unfolded region. So they made a DNA RNA hybrid. So this DNA sequence is complementary to the strand, flow across the switch sense, and you could see then the association of just the domain by itself, and then the one with the upstream unfolded region. And this association, so you're decreasing the dynamics of that switching. And then when you flow buffer, you could see there was quite a big difference, if you can see, in terms of the dissociation time. With when you had the upstream region, there was much, much slower dissociation off. And so then this upstream region, then it's increasing the affinity right by slowing that off rate back. For those of you who were at the fluorescence 1 lecture, then you'll remember this. Again, example here. And they wanted to look in more depth about how this RNA binding protein here led to specificity of targeting this deadenylase complex here. What were these sequences here? How did they affect the kinetics of binding here? So you could see that one type would be specific for its sequence and would be how much higher affinity compared to the other sequence, or just poly A. And so again, they made then the sequence here where they would make a DNA and RNA hybrid, which would then they could put across onto switch sense. And they also included then a mutation here between an aniline then to uracil, and looked at the differences. And they could see that, well, if you flow across the one that's targeted to the puff free, you can see this is the on rate. It's much, much faster than, say, the off target. And if you changed then the adenine to uracil, then you slightly changed then the binding. But really this on rate for the sequence was about 7-fold above just poly A. And if we look at the dissociation, then what they could see was that with poly A, it really, the puff free just dissociated very, very quickly, really. I mean, you know, compared to then say, you know, say an off target, or if you had uracil there in that sequence 5, and then it was much, much slower off if you had the correct sequence. With then switch sense, then you can do other things. I'll just briefly skip through this. Because you can look at polymerases, then you can look at on rates, elongation rates, and the extranuclease rates, and also the dissociation rate as well. And there's, if you go to the company website, there's lots of examples there. And so you can actually tease out all this different kinetic rates. And you can do this at different temperatures as well to look at the thermodynamics and also build up a mechanism capping. So I'll leave it there for today. And if you want kind of more information, for those internal to the LMB, you can look at the internal biophysics website. We've got lots of different information on different techniques. For people external, then there's the company websites. For SPR, then there's this SPR pages, and obviously for switch sense and Octa as well. These are the company pages as well. So sorry for going slightly over time, but now, you know, thanks for staying here. Thanks for your attention, and obviously I'll take any questions. If you want to write them into the Q&A box at the top there, then I will answer them. Okay, so a question here from Sissy asks, does the surface charge of the protein influence the switching? I would say that probably not, because with the DNA, then you've got, you know, quite a high charge on that surface compared to a protein. But obviously, if you say you bind a protein, say, say it's a DNA binding protein that binds onto the DNA, then obviously you're going to mask that charge. So that will actually influence in switching. But if, say, the protein then binds onto the top, say to something attached onto the end of the nanolever, then I don't think it's going to make that much difference. Okay, George asks, he wants to study a DNA binding interaction. What's the size range of DNA that can be used? Well, I mean, there's two types of DNA then levers, and there's a 48 and there's also a 96. But you could also, you know, obviously extend that DNA region by then having, say, both a single strand that then goes beyond the nanolever then and forms a duplex with your strand, which will go beyond that. But obviously, you know, you could have, say, a specific sequence beyond the nanolever, but you obviously will reach a point where the switching probably won't be as good, or you get some more flexibility in there. The question would be, you know, how long do you kind of need it to be to look at the interaction? But we have done, you know, lots of experiments where we have had DNA on the top, and you've looked at interaction. Okay, if there's no further questions, then I'll say, you know, thanks very much for coming and staying with us today. And then don't forget, then there's then the next talk, which is going to be on Tuesday, I think. Please come again, and if you can't come again, then have a look at the YouTube channel.